really been fantastic for us to be able to get in front of the broad audience that have come together this morning and to be able to listen to, I suppose, the story of where the hub came from, what it's doing, and what we hope to do in the future. So thank you very much for that, and I hope that people find this an enjoyable presentation with lots of information about the broader scope of what we're doing in the hub outside of those healthcare projects. So as Emer said, I'm the Associate Dean for Research in the Faculty of Science and Health. My remit in that role is really about the coordination and execution of the research strategy for the Faculty of Science and Health. It's one of the most research active faculties in DCU. I'm also a professor of immunology, so I have a great understanding of what's going on in the COVID-19 space at the moment, because it is slap bang in the middle of my own research expertise. So what I wanted to talk to you about this morning was where the hub came from, and what the goals of the hub are, and why we got together in the first place. So one of the things that was very clear when this kind of pandemic really came to the fore, late March of this year in the country, was that DCU staff were almost putting their hands up to ask what they could do to be able to help. So we had lots of things that happened across the campus. We became one of the national call centres, and we had staff that volunteered to run that call centre. We had the establishment of the COVID-19 assessment hub with the HSE, which means that any of the patients that have to come in that need a medical assessment by a GP avoid the GP surgery and go basically straight to that assessment hub. And then we started to do things like 3D print visors for healthcare workers, and we started to reach out to whoever our collaborators or stakeholders were that we were working in research and ask the question, what can we do? In parallel with that, some of the national funding agencies started to put out calls for funding for research projects which would address some aspect of the current pandemic. And because our staff were literally chomping at the bit to be able to help, we decided that we needed to be able to put together a very coordinated approach to our efforts. So the academic community and its research strengths is really what this DCU COVID-19 hub brings together. We recognised very early on, and the input from our academic researchers was that while the initial, I suppose, public awareness was around things like testing and trying to get better tests, and one of the things that we were very aware of was the fact that the impact of what we could do would be much further than just that technology piece, and taking our staunch kind of hard science and engineering expertise and applying it there. So we had people from the humanities and social sciences, we had people from the education and the business schools. And really what we did was we decided as a university, very much led out by our president who just recently finished his term, Professor Brian McCrath. He started conversations with the DCU Educational Trust. And that Educational Trust very much came behind us in terms of being able to put funding behind us to get some of the research projects started. We put out a call to the university, got 75 applications, and we managed to fund 15. And we funded those broadly across all areas. So that one of the things that we were very conscious of in that call was that whatever people were going to propose to do, it had to have as much of an immediate impact in the current pandemic as possible. So the technology projects that we looked at, a lot of them were about whether or not they could achieve what they set out to achieve within a 3 to 6 month period. A lot of the economy and society projects, and I'm just going to give you a snapshot of a few of those, it was really about trying to impact on an individual during the current pandemic, whether it was trying to create solutions around how people in society were managing the pandemic, or whether it was solutions in terms of diagnostics or therapeutics. So we had a very, very broad remit. So with that, with the very wonderful support of our DCU Educational Trust and the full support of the President of the University, we established the COVID-19 Research and Innovation Hub. We seed funded 15 projects across the full breadth of the university. And then what's really nice is that not only does the hub give us a way of coordinating internally our efforts, but it also gives us a way to externally communicate what we're doing in this space. And we put extra support behind our researchers in terms of trying to leverage what we had established in the hub to apply for further funding. And we've been very successful to date in terms of that we've leveraged a lot more funding from Science Foundation Ireland. They had a very specific COVID-19 rapid response call. HRB had the same, and very recently the Royal College of Physicians of Ireland. So we have leveraged funding across not just the health sciences, but also engineering, humanities, and social sciences from these funding agencies. So our hub has grown from our seeded funded hub, the Educational Trust, into a larger entity, which is now winning competitive funding from several funding agencies. And apart from these three, we also have a number of other pending applications for leveraging funding. So we've really grown in the last 4 months that we have been established. Just to kind of give you a snapshot, like because of the audience that we have today, when we had some discussions with Emer, we talked about presenting a couple of projects that would be relevant and that there might be some interest in. But I wanted to just kind of give you a little whistle-stop tour just around the other projects so that it's very clear that there's, there's breadth to what we do, that we are interested in projects that impact on all aspects of society. And that's what our goal is to be able to impact across the totality of society. So for example, in terms of our technology projects, these are really focused on leveraging the technology that we have and applying it to use as solutions for problems that have come up during the pandemic. And obviously as a university, one of the key issues that we are very interested in at the moment is about how to enhance learning online and how to be able to navigate through that very rapid transition of putting our teaching online, which we had to do literally overnight back in March and April, and have had to do all summer for the coming semester. So, for example, Professor Mark Brown leads out on a technology project that is very much focused on the students. So understanding where the issues were in their online learning, correcting those solutions, and putting a MOOC online, which is like a mini online course, so that students can access that in order to be able to educate themselves and enhance their own learning through their knowledge of the way technology is being used. The other issue that we had was very easy for us to be able to do online lectures for small classes, but it was a real challenge for large classes. So, for example, one of the modules that I would have taught would have been biology and cell biology and biochemistry for first years. And there's approximately 280 students in the class. Now, while Zoom can hold that number, it doesn't really lend itself very well to interacting with the class and getting feedback. So it's very difficult to manage large class sizes. We would have quite a proportion of our class numbers across the university, which would have large class sizes. So Anne-Marie Farrell leads out from our Institute of Education, which is, as you know, the only one in the country. And really it was about trying to maximise what we do for our large classes online and being able to come up with a package that we could communicate that to the students. And the outputs of this project now are being translated this very month so that we can start to implement those solutions for the first years, particularly that we have coming in on the 5th of October to the university into a very different environment than we've ever had first years coming in before. Just to change tack then a little bit, we have a very well established School of Business at DCU. One of the things, two of the areas that we have a lot of expertise in is the area of HR and the area of working with frontline workers. So we have a long history of working not with just nurses and doctors on the front line, but we have lots of relationships with both the Guards and also Dublin Fire Brigade. So again, harnessing the expertise that we have, Professor David Collins led out on a project which is really about understanding the key challenges that human resource leaders had to deal with during the very quick change to working at home and the change in the working environment for their staff. And really the objective of this project is to be able to garner that information and be able to come up with solutions so that we can start to inform HR directors out there in our corporate partners or our corporate clients and start to inform them about how better to be able to manage those challenges into the future by learning from those who have figured out these solutions and put things in place that have worked for them. The Listen project, again, was about understanding the challenges that the frontline workers were dealing with and being able to, not just in this pandemic, but in future crises, being able to try and come up with solutions, whether that is peer support, whether that is access to particular resources that they would have found would have helped them at the time. It is really to inform how we move forward with this pandemic and also future crises or pandemics. In terms of looking at our societal impact, one of the key research objectives of DCU is to translate our research to be able to impact societally. One of the things that arose during the initial stages of the pandemic was that there was the communication was predominantly given the country we're in was in Irish, and in English, excuse me. And one of the things that we know, obviously, the way Ireland has developed over the last 20 years is that there are an awful lot of minority groups who speak a different language. So one of the things that we feel that led to was, if the translation wasn't right and the information wasn't right, whether you could trust the information. So really it was about looking at minority groups and looking at how did they get the information and how did they, I suppose, understand that information and whether or not it impacted on their engagement with, say, with public health guidance, for example. So that was a really important issue that we felt needed to be addressed. And we actually had a separate project that addressed solely the Irish language and the translation into Irish of the information that was coming out. And then Jane Souter from the School of Humanities and Social Sciences, again, understanding a little bit more about how citizens responded, what their attitudes were, how they changed their behaviour when the environment that they were living in changed. And again, trying to figure out how did that impact or influence their engagement with public health guidance and how did their behaviour change according to the information that they were getting. So Jane Souter is very well known for her citizen science project and citizen assembly projects, and she would be very much an advocate for citizens having a voice at the table in terms of how decision-making processes are moved forward, but also about policy development. So she was a great person to lead out on that project, and the information that they're getting in, we feel will really help us in terms of trying to understand better how people respond and behave, and then what we can do to be able to take advantage of that. And then switching a little bit towards huge issues around how our children, I suppose, dealt with the pandemic, and a very, very strange situation for them all to be in. Lots of you will have seen an awful lot of stuff that came online in terms of physical education at home, and the PE stuff that was done online and on the telly. One of the things that we did with this project was we brought together our expertise in health and human performance and our expertise in education. and we developed a PE program that was age appropriate, and it was to do with developing skill sets. So it wasn't just about keep fit and get the children moving and being active. It was more about taking a set of skills that children needed to have at certain ages and developing an at-home program around those. So those were rolled out, and actually the stage that that project is at now is actually doing the assessment of the impact on the skill sets development for the children over the lockdown. That's been done predominantly through their parents. So that project is quite advanced. And then finally, just mentioned one of the projects there that we leveraged funding from through the Health Research Board. And it really addressed a very, very important issue around children with special needs that didn't have access to their normal supports during lockdown. And autism is Dr. Sinead Smith's area of expertise. And it was really taking and looking at the experiences now of families and parents dealing with children that have autism, how the lack of structure and the change in their life has impacted. And they're now taking that information, putting together a set of resources that will support those children at home, and more importantly, support their parents in terms of keeping the children at home, but also being able to manage the fact that they're not engaged in their normal resource supports. So a really important project with very, very important impact. Okay, so they're just a snapshot of some of the projects that we're doing across the entire breadth of the university. You'll see that the expertise that we bring together is very broad, and the impact and the solutions that we're trying to develop are very varied. We funded 4 projects in the area of healthcare. And again, we were very focused on what was very topical at the time, which was about trying to improve the testing, but also to be able to try and get a handle on what was happening with the things like the antibody space. So, for example, we were getting a lot of stuff was coming out in the media. New companies were launching new antibody testing the whole time. And there was pros and cons with each of the tests. And one of the things that some countries, including Ireland, were very reluctant to do was to, decide exactly what kind of a test they wanted to use. We were plugged into the HSE Research and Solutions Group through one of our principal investigators in the university. And essentially through that group, we developed this project where a number of sites around the country linked into clinical partners would essentially take some of these tests, some of which are lab-based and some of which are point of care, and that they would do a full assessment of which ones would be the best ones to use for monitoring the antibody responses of the Irish population. So that is a project that is in process at the moment with a number of clinical partners and a number of other universities. And the objective is that the information that comes out will be fed directly back into the HSE and hopefully inform their decision about how useful antibody testing will be going forward, but also about which are the right tests to use and which ones are giving us the best reliability. So the independent validation of those is very, very important. Another project, Professor Stephen Daniels in our Faculty of Engineering. So one of the focuses has been on the testing, obviously, of individuals to look for the presence of the virus. One of the other things that we know are that transmission of the virus through surfaces is a real route of transmission, and one that causes a lot of difficulty. And we also know that monitoring healthcare settings for any presence of the virus is absolutely critical to us moving forward in terms of maintaining our healthcare settings to be free of the virus. So testing individuals is one thing. So Professor Daniels is developing a low-cost device which will detect COVID-19 on surfaces in healthcare settings. And based on this project, he managed to leverage funding from SFI where he's now applying similar technology, but for the detection of COVID-19 in the environment. And Professor Daniel's expertise is in environmental monitoring of pathogens in the air and on surfaces. So this is perfectly matched to his skill set, and we think a really important project for being able to monitor individual wards, entry points, exit points of the hospital, and making sure that surfaces are kept clean, but also that the presence of the virus on a surface would be able to give us an early indication that there's potential for the virus to transmit in that setting. And one of the ones, obviously, that we're very interested in is nursing homes. And then the two projects that I'm just going to, I have a couple of slides on each project just in terms of giving you a little bit more detail. One of them is about trying to, we phrased it the shrinking of the COVID-19 assay for faster results. It's really about trying to address the issue of the length of time it takes for testing. And I'm going to talk about that in a little bit more detail now in a moment. And then the second one is, while there's a huge focus internationally on the development of vaccines, one of the important interim measures is to find therapeutics that really treat severe disease. And we've heard a lot in the media about trialing of different drugs that we think might be able to interfere. There's now a full panel of these, everything from antivirals to anti-inflammatories, and the anti-inflammatories really are addressing the inflammation, the chronic inflammation that happens in some individuals as a result of the virus being present, and that's very much down to the individual involved, and there's no consistency between the kinds of symptoms that everybody gets, because everybody doesn't get respiratory inflammation. So one of the things that we had was Professor Hughes. Sorry, it's just a bit of noise. Is that gone now? Yeah. So Professor Keyes actually has adapted a novel platform that she has developed, and I'm going to talk about how she's adapted that in order to be able to provide the pharma industry, essentially, in the first instance, with a way of rapid screening of any small molecules or bioactives or peptides to see if they block the entry of COVID-19 into cells, specifically through the receptor that we now understand a huge amount about. Okay, so I'll just spend a couple of moments on each of these just to give you a flavour, I suppose, of what's involved in these projects. So Dr. David Kinahan is based in the Faculty of Engineering and Computing, as is Professor Nick Dunn. And Professor Helen McCarthy is a new international adjunct professor that we have recently recruited to DCU, and she's based in Queen's University. And really the focus here is about taking the time issue, but also taking out the amount of input from a technical person in the lab in order to be able to carry out this test. So it's very much about trying to take the swab, putting it into a closed assay system that will not only work fast, but that once the assay is closed, then there's no more involvement of putting in different reagents or progressing the assay until it's finished, which will take about 15 minutes. So it's a low-cost automation of molecular diagnostics assays. Essentially, it builds on, as do all of our projects, they build on an expertise that we already have in DCU, and we're just applying it to this new problem that we have. It builds on DCU's expertise in centrifugal microfluidics, and this microfluidics really grew out of the expertise that was developed in the Biomedical Diagnostics Institute, which is one that was funded by Science Foundation Ireland in the past and led out of DCU. One of the things that has come out of that in the last couple of years is that we are one of very few countries outside of Germany, and the only one in Ireland, the only university in Ireland to have a Fraunhofer centre based on our campus. And the sole expertise in that Fraunhofer centre is around microfluidics and the exploitation of microfluidics in order to be able to develop rapid diagnostics for an array of different disease states or detection assays across a number of sectors. So really this is about trying to make a point of care test by using the microfluidics expertise that we have, but specifically centrifugal microfluidics, which essentially means that if you think about a centrifuge spinning really, really fast, the way our discs are made in order to be able to do this assay is that the centrifugal force actually mixes all of the individual parts of the assay while the disc is centrifuging around. So in other words, once we put the sample into this system and use centrifugal force, the different steps of the assay happen themselves. Okay, so it's a really, really clever assay. So we've basically adapted this platform, or we're in the process of adapting the platform to provide low-cost platform which is compatible with swabs or saliva samples. So we have another project that is more specifically based on saliva samples that is that's pending for competitive funding at the moment. And this one really will focus initially on swabs before it moves on to saliva. It's fully closed loops, hands off, load the sample, put it onto the centrifuge holder, which is has an arm. It's a platform that's been developed specifically for this, and the report answers into a smartphone app. So it's really about trying to automate as many steps as possible of this and to minimize the time. One of the other things is that the cost of this is very, very low compared to the normal cost of a test. So in terms of the workflow of this, it is about having the microfluidic chip, which is what you load in for the centrifugal force to be able to mix the different parts of it. You put in your swab, it's scanned to a barcode that's on the chip, which will have all the details then of the patient number. And then that's all fed in essentially once it's centrifuged, and the detection is read by a reader instrument and the information is stored. So it's supposed to be an end-to-end rapid assay, really taking out the need for involvement of a lot of man hours. And as you know, one of the things that's come out in the news in the last few days and certainly been shared a lot on social media is about the job that the technical people have done in the labs, in the clinical labs, in the virus reference lab, in places like ENFR, that they have done for the last 6 months continuously, that the resource, that they're getting tired, and that the resource needs an influx because it's going to be really hard for them to sustain that, particularly through the winter months. So the advantages of something like this, it's very compatible with low-cost manufacture. One of the things we have in the Fraunhofer is the ability to be able to manufacture prototypes, but also through our partnership with our German partner is to be able to scale that up. So we have the ability to be able to do that once this works. It uses LAMP technology, which is different to PCR. So it's a different type of technology, uses isothermal amplification chemistry. So it's not the PCR that we use and the method that we use, that labs are using now. It should be suitable for a range of samples, including swabs and saliva. Can be easily adapted to support emerging tests. And what we mean by that is that if another virus should emerge in the future, that we will have this platform set up to be able to apply to that. And it has the potential to be able to provide an answer in 15 to 30 minutes. So the turnaround time is very, very quick. The video, I tried this earlier, but it didn't work, so apologies. It showed the disc on a centrifugal arm and basically rotating. And this rotation, I'll show you now on the next slide. This is essentially what a disc looks like for an assay. And essentially in each of these parts here will come fully loaded with whatever part of the assay is needed, whatever reagents, are needed in each step of the assay. And it is the change in the centrifugal force that mixes different reagents with the sample at different times in order to be able to give the readout. So it's a very clever system that we've used a lot of times before. And the principal investigators that lead out on this see this as being something that can be used in COVID-19 testing centres with very quick turnaround answers. It is something that can be used at airport screening, which, as we know, is an issue at the moment. Hospital A&Es, and one of the other things is to be able to be cognizant of the fact that while we're very well equipped in lots of countries in Europe like ourselves to be able to have lab facilities, not as easy in developing countries to be able to have not just the resources and people with expertise to be able to run these tests, but in developing countries, sometimes these quick turnaround, cheaper tests is more conducive to their lack of technical support that they have. And then finally, just on to one of the other healthcare projects that we have at the moment. And Professor Tia Keys has expertise in developing platforms that look at interactions. Okay, so essentially this MEMSense platform that she has developed previously looks at, it basically represents a full bilayer membrane. It is stable, it has the proper membrane lipids in there, the diffusion of proteins and other substances through the membrane is possible. And so it's a very highly ordered membrane. And one of the things that it's been used for in the past is being able to mimic the attachment of things like influenza virus, for example, to cells, and look at that attachment and that initial interaction of their entry into cells in more detail. So that's essentially the platform that she has. In terms of binding to the platform, there's a number of different ways in which that is measured, okay, and usually lasers are the kind of first port of call. So really spectroscopy and fluorescent microscopy are the ways in which any interaction with that membrane is assessed. So she came up with this idea that she would modify the membrane that she has in order to be able to incorporate the receptor that the spike protein on COVID-19 binds to. So the ACE2 receptor, as we know, is one of the key points of entry into a cell from COVID-19. And therefore, one of the first ports of call for an antiviral is to block viral entry into cells. So anything that can block the interaction between that spike protein and the ACE2 inhibitor is potentially an antiviral that would have use against COVID-19. So essentially what she's done is she's incorporated the ACE2 receptor into her MEMSense platform and is now in the process of being able to confirm that the COVID-19 spike protein interacts with the ACE2 receptor, and to start looking at whether or not this interaction can be blocked in any way. So the end goal is to be able to have this model set up, have it validated, and then if there was companies, for example, with large libraries of peptides or bioactives or anything they think might be useful in this space, that it would become a screening tool, essentially, to be able to look at that. And the other thing, obviously, it does as well is that it actually gives you a really nice model for us immunologists, but also biochemists, to look more closely at the interaction between the glycoprotein, the spike protein, and the ACE2 receptor in order to understand the virus a little bit better, because it's so new and there's still so much about it that we just don't know. So essentially, incorporating the ACE2 into the membrane has already been done and has been shown to be successful. The second part that they've moved on to, that they've also had very promising results from, is to be able to look at when the spike protein it attaches to the ACE2 receptor, are they getting a measurable signal by either electrochemistry or fluorescence? And then the next part of that is to look at the affinity of ACE2 and spike in order to be able to understand better the interaction. But then once that's set up, it'll be available to test a range of drugs for inhibition of that attachment. And she had just included one of the preliminary results in terms of being able to demonstrate that that you can measure the interaction between the ACE2 protein and the spike protein. So, for example, where the ACE2 protein is not present, in other words, the spike protein doesn't have anything to attachment, our measurements are very, very low and they're almost on a straight line. And when you actually have the ACE2 present, and these are two different conditions, and what you see is that there is an increase in the signal, which means that the interaction between the spike protein and the ACE2 protein, the ACE2 receptor, is taking place. So those are just two of the projects that we're looking at in detail, and I hope I've given you a very broad flavour of the rest of the projects. And I suppose for us in DCU, we're really conscious that we want people external to the university to be aware of what we're doing, to understand better what we're doing, and if there is, any kind of overlap of areas of interest, that it might be a start of a talking point in order to see whether or not there are any kind of areas of complementarity between external stakeholders and DCU. So thank you very much for your time. Thank you for listening. Jennifer Hoey is one of the lead representatives on this project at DCU Educational Trust. And, if there was anything else that you wanted to understand better or know about the hub, you can follow up with Jennifer. And then I've also included just a link to the website, which gives a bit more detail on the COVID-19 research and innovation hub. So thank you very much for listening.
That's great, Christine. Thanks a million for that. I think Oliver O'Connor should be on the call here as well. And Oliver, I think, is going to pop on to say a few words.
Yeah, thanks, Emer. Thank you very much indeed, Christine. And first of all, apologies that I was not there at the very start of the presentation, which I should have been, but that's just my own fault. I've no excuse to offer. I'm sorry I missed the first 5 minutes because I was really keen to see this session and in its entirety. But I was there for nearly the entirety of your presentation, and I really found it very interesting as a non-scientist indeed. It is very impressive what DCU is doing now across the range of research that you're involved in, not just in the technical parts that you have explained there in the last few slides, but across the social sciences as well, and the importance of all this. And it is important for us in IFA that among our member companies and indeed our affiliates, that we too would understand what is going on in DCU, what research you're doing, and indeed the contribution that you're making towards science in this area, because obviously, our member companies are focused very much on scientific developments and solutions, particularly in the COVID context. So we're delighted to have an opportunity to provide some briefing like this, even to a lay audience, as we explained in our call yesterday, and those who are slightly more than lay, of which some of the participants are, will have understood in detail the last few slides that you've mentioned. I think it's, speaking for myself, I think it is really good when we see a rigorous scientist like yourself describe a development as very promising, to do with the ACE2 receptors and so on that we've all been reading about. That's really good to see. If you don't mind, I will start with a question. I know we have the facility, Emer is going to take questions on the chat, but I will start with a couple of questions, if I might, to you, Christine, if that's okay.
Yeah.
To say that has occurred to me in looking at listening to your presentation. And the first ones are really to do with the context and speed. So in COVID, of course, as everyone knows now, pharmaceutical companies research institutes, academia are racing at breakneck speed, unprecedented speed, to develop both vaccines and indeed therapeutics for COVID-19. How have you experienced this question of speed in work in DCU, and also the commitment indeed that the pharma companies, and I'm sure academia will make as well, to say, look, we have to keep up standards of integrity for the research, because it really ultimately is of no value to anyone if in the rush for a solution we compromise on those things. So how have you experienced that in your work in the last number of months?
Yeah, no, it's a really good question, especially with the news this morning that one of the COVID-19 trials has been halted out of the Oxford vaccine because of an adverse event. And people are now kind of asking this question about the speed at which we're doing things. I guess that one of the considerations that we made when we chose the projects that we were going to do was that we prioritized the projects where it was built on an expertise that was directly applicable to another area, so that none of these projects are starting from scratch. You know, they're not ideas where we're right back at the start creating platforms. They are about our low-hanging fruit. They are about saying, what do we have already that might apply to this area? So we feel like we've tried to kind of overcome that potential pitfall of speed by making sure that anything that we're pushing forward has like decades of research expertise and validation behind it, so that we know that platforms work, and really it's the changing of that platform and the application to a different space. But one of the things that we laid down was that the outputs of these projects had to be very tangible within a 3 to 6 month period. Now, the 6 month period was more applicable to the healthcare ones because they are harder projects. And one of the challenges that we had was the lengths that the university had to go to be able to allow access to researchers to come back on campus and actually start their COVID-19 work. And that was very much a process that happened during April, May and June to be able to physically get researchers back on campus. So we were one of the first universities, and we were certainly the first faculty to have our researchers back on campus, and it was purely for COVID-19 research. So we have tried to be able to overcome any of the issues around the timing, but we are very confident that, you know, by the end of Q4 of this year, that we will have tangible results that can be potentially directly commercialized. You've already seen that with Professor Key's project, that there's a huge amount of that project already done by virtue of the fact that the platform was already there and validated for a different interaction. So I kind of feel like we've been very cognizant of those things to make sure that we do things that are attainable and are realistic, and do what we can to overcome any of those challenges. But certainly the biggest challenge that we will have is the amount of time that it may take to commercialize some of these.
I see. And has the speed of funding increased in terms of has that responded in terms of to the needs of the situation?
Yeah, so I mean, one of the things that happened was there was an initial, I suppose, drive from some of the funding agencies to try and fund COVID-19 research in general in Ireland. One of the things that I will say is that the DCU Educational Trust trumped all of that by providing seed funding before any of those calls ever came out. It was a very, very quick process that we did internally. And that's why we've been able to leverage further funding, because we already had those projects in place, and we've just bumped them up, if you like, and expanded them by getting additional funds. But what I will say is that SFI, HRB, you know, the RCPI, there's been lots of calls out. There's been lots of European calls out. One of the things that the national funding hasn't addressed is the scale of funding that really is needed to be able to address something like this. So some of the projects that are funded externally may be into the 100,000 or 200,000, but when you look at the extent and the cost of the pandemic to the country, the amount of funding that national agencies have put beside it is minuscule. And that is one of the biggest challenges that we as a research community have, and we have it in DCU, is that the desire and the graw and the absolute determination to be able to apply our knowledge to finding solutions is there. The biggest issue that we have is that the funding is not where it needs to be.
Interesting. Okay.
So our audience are being quiet this morning, and there hasn't been any questions loaded up on the chat there. I suppose my main question to you, Christine, is where do you see the future of the hub going? You know, right in the middle of the pandemic, it was necessary. Right now, you obviously have a lot of projects going on. Is this a long-term innovation that you see? Is it just, or does it all depend on the course at which COVID takes? I suppose what's your view on that?
Yeah, and that's something that we have discussed quite a lot in the last couple of months, because depending on what report you listen to, COVID-19 may be with us for quite some time. Or, if a vaccine breakthrough comes, it may be kind of a distant memory, hopefully in the next year or two. However, I suppose what it has done for us is that it has highlighted that when faced with the crisis that really needs science to try and solutions, but also society to change behavior, that we need to understand more about how we do that so that we are better prepared next time. So our kind of longer term view of the hub is that it will become hopefully sustainable through partnerships with external stakeholders that see the value in what we do and realize that their long-term investment in something like this will be very valuable for the future. But really, our expertise that we're applying in this situation, it is the same expertise that we need to be able to make sure that we have and understand to be able to apply to other crises in the future, and not just another virus, but anything that comes along that radically changes the way that we live very quickly. So a lot of the projects that we didn't fund had longer term kind of goals. So for example, understanding how different areas and cohorts in society responded to the public health guidance, understanding the impact that it had on cohorts, everything from the kinds of communities that we work with in DCU, everything from the homeless community to people with special needs, to healthcare workers, children, vulnerable adults, those living with chronic disease. And the problem is that we didn't have any preparation for this. So everyone was told to do the same, and the services for everybody was pulled. And really what we're trying to do now is retrospectively understand the impact that's had on people and their disease state, their mental state, etc., and come up with an informed set of solutions that will help us respond better the next time, or help us respond better next year if there's a second surge, for example. So the understanding of people's experiences and behavior and the impact it's had on them is a really valuable piece of information that we actually need to know for the next time. So we would see that our Research and Innovation Hub is directly applied to COVID-19 at the moment, but the longevity and the sustainability is around crisis preparation and understanding how we address crises in the future.
I suppose just from an academic point of view, you mentioned at the beginning of your slide deck there about how all classes had to go online, everything. So you had to change so quickly. Do you see a change in the interest, I suppose, increase in interest of incoming students in this area? How do you see the impact of COVID on DCU as a learning institute? It's fascinating the fact that you had the humanities, the business, the science, all merged together in this hub. So it's obviously going to have a follow-on impact on all these different faculties as well. And do you see a big change in how courses are going to be taught and content, etc.?
Yeah, I mean, it's a real challenge for us to move into next semester with kind of the understanding that the majority of everything is going to be online now. And that's a massive change for students. Some of our students, our continuing students, obviously have had experience of last semester and everything being online for half of that semester. They will adapt quicker. Our first years who've come in from a school environment, the engagement, physical engagement with the lecturers and their peers is a huge part of what we can't replicate online. And that's one of the things that's happening with that large class size, is about how we manage to do breakout rooms and smaller class sizes, and how do we get them to engage with each other. So there's a non-scientific part of what we're trying to do now with our students, which is actually about their experience. And while DCU has always been very focused on the student experience, this is another level for us in terms of how we use our technology to try and recreate that experience as best we can. In terms of the faculties working together, you know, this is something that has kind of, we've always been known for our multidisciplinary research. We've always been known for being able to kind of pull a diverse group of expertise around the table and kind of thrash out a problem that uses like a different and diverse expertise. This hub has been another way to be able to drive interactions between those faculties. And one of the potentials that will now happen out of that is that, so if you just look at the example I gave you of the health and human performance and the education, it is likely that that will start to emerge in our curriculum. So anything that we do in terms of our engagement with research does start to emerge then in the curriculum as a result of that. And that could be a real positive for our students in terms of the kind of experience that they get and the mixture of disciplines that they get expertise in. So they may be, you know, very different coming out in 4 years' time as, you know, as they are coming out now. I think we're going to be looking at a very different student over the next few years. But we are worried about the student experience, and we are doing everything we can to mimic as much as we can. I mean, in Faculty of Science and Health, we're still having our students come on for practical lab-based work. And we've managed to work it out so that we can do it in a safe way. And it's been a mammoth task. But we're very conscious that DCU is known for its excellent laboratory skills and the training of the students, and we can't let those standards drop. So we're adapting all the time, and it's been a pretty tough year to be an academic working in the university, as I'm sure it's been for every profession. But trying to engage the students and keep them excited about what we do is a big part of it.
Excellent. Well, it is now 11:59, and we said we'd keep everybody for an hour. So I think on that, we might wrap things up. Christine, that was super. Thank you very much. I think
Thank
everybody
you.
here across the board, scientific and not, would have found that of great use. I will, as I said at the beginning, I will circulate the slides and the recording of this. And more importantly, if anybody's any direct questions, to contact you guys there at DCU on the contact details provided. So on that note, Oliver, I'll bid everybody goodbye.
Thank you very much indeed, Christine. Thank you, Emer, and thank you for the participants, and we'll be talking more.
Great. Thank you for your time.
Okay. Thanks, Million. Goodbye.
Bye.